Hello everyone. Today we are going to study a new section, the cell injury. We all know that if the stress exceeds the cell's adaptive capacity, or the external stress is inherently harmful, then cell injury develops. Cell injury is a reversible change and culminates in cell death. The causes of cell injury can be summarized as follows. Let's go through them one by one. Oxygen deprivation. It is a common cause of cell injury and death. The cause of oxygen deprivation include the first, ischemia, because the arterial flow is impeded or the venous drainage is reduced. The second point, heart or lung failure, which will cause inadequate oxygenation. The third, reduction of the oxygen carrying capacity because of blood loss, anemia, or carbon monoxide poisoning. The second cause is chemical agents. An enormous number of chemical substances can injure cells. For example, poisons such as insecticide, ethanol, air pollutants, and so on. They are potentially toxic agents. Even some simple chemicals, for example, the glucose or salt, in hypertonic concentrations can also lead to cell injury, even death. The third cause is physical agents. Mechanical trauma, extremes of temperature such as burns or exceeded cold, changes in atmospheric pressure, radiation, electric shock, they all have wide-ranging effects on cells. The biologic agents, including viruses, bacteria, fungi, and parasites. The fifth cause is immunologic reactions. Firstly, anaphylactic reaction to foreign protein or drugs, for example, food allergy, can cause cell death, rashes on skin, and tissue edema. Secondly, autoimmune reactions induced by endogenous self-antigen. The self-antigens can cause many diseases such as nephritis and hyperthyroidism. There are some other causes, for example, the genetic defects, nutritional imbalance, endocrine aging, and so on. The mechanisms of cell injury is for self-study. Okay, we see this page again. Cell injury consists of reversible injury and irreversible injury. The former, also named as degeneration, the latter leads to cell death. We will talk about the degeneration first. This is the definition of reversible injury. The metabolic disorder will damage cell or extracellular stroma. Then, morphologic changes will occur. Some abnormal substance or excessive normal substance will deposit in cell or stroma. Then you might be curious about the substance accumulation in intracellular or extracellular space. Well, the excessive normal substance in cell or stroma could be water, lipid, protein, etc. As for the abnormal substance, they could be infectious products, mineral substance, and so on, which could be endogenous or exogenous. There are 9 types of degenerations. We will talk about them in detail. The first type, cellular swelling, also called hydropic degeneration. It is the first manifestation of nearly all forms of injury to cells. The common sites are liver, kidney, and heart. It is usually caused by hypoxia, infection, and intoxication, which finally leads to the mitochondrial damage. And then the ATP synthesis decreased. Sodium-potassium pump is dysfunctional. Then water-sodium retention occurs. Pathological changes of hydropic degeneration. In gross appearance, the weight of organ is increased with tense capsule and blunt edge. In the cut surface, the color of organ become pale. Okay, let's take liver as an example. This is a normal liver. This is a cellular swelling picture. We can see in the second picture, the swelling cell is much bigger than the normal one, and they have lighter color and lots of tiny red granules in cytoplasm, like here. In serious hydropic degeneration, the third picture, the swelling cells are huge and round. For example, this one and this one. The cytoplasm become very clear, looks like empty. We call this as ballooned change. The second type of degeneration is fatty change, also named as steatosis. Fatty change refers to abnormal accumulations of fat within parenchymal cells. The common sites are liver, heart, kidney, and muscle. The major causes of fatty change are hypoxia, infection, intoxication, and obesity. Fatty change is mostly found in the liver. It may be caused by toxins, protein malnutrition, alcohol abuse, diabetes, obesity, and so on. Alcohol abuse and diabetes associated with obesity are the most common causes of fatty change in the liver in industrialized nations. Here we have 2 pieces of liver. The left one is fatty liver, the right one is normal liver. Compared to normal liver, the fatty one is yellow and soft. The cut surface is greasy. In this picture, the fatty liver looks smaller than normal one. That is not true. You cannot judge the total volume by one piece of the liver. In fact, the fat liver always have a larger size and weight than normal liver. Under microscope, the fat vacuum is round and empty. Some of them are large, like this one, and some of them are small. And please look at this cell here. The nucleus and the remaining cytoplasm are pushed to the peripheral zone by the huge vacuums. So makes this cell looks like a fat cell. You know, the fat is dissolved by organic solvent during section processing to leave the empty vacuum in it. Sometimes we want to make sure what is in the vacuum. Then frozen section with Sudan III staining can tell us the answer. The fat show orange-red color in the section.